I want you to picture something that's become just frustratingly common. You get COVID, the usual thing. Cough, fever, you feel awful, you do the right thing, you isolate, and after a while, you finally get that negative test.
Cue the relief.
Exactly. You think, great, I'm done, I'm in the clear. But then, well, the timeline gets weird. A month goes by, maybe 3, and your stomach just starts acting up. I mean, really acting.
Talking chronic stuff, right? Not just a bad meal.
Oh, yeah. Chronic diarrhea, abdominal pain, reflux, the works. So you go to the doctor, obviously, convinced something is seriously wrong. They run blood work, they check for an active infection, and nothing. Everything comes back clean.
And that is the classic ghost in the machine scenario. The map says the territory is totally fine, but you, the person living there, the ground is shaking under your feet.
It's like being gaslit by your own body. And that ghost, that glitch, is exactly what we are unpacking today. We're diving into a paper that just came out, March 2026. It's called Impaired VLCFA-peroxisome-mediated intestinal epithelial repair causes gastrointestinal sequelae of long COVID.
And I know that title is a mouthful, but please don't let it scare you off, because this is honestly one of the most complete molecular detective stories I've read in a long time.
A detective story. I like that.
It is. It traces the crime from like a huge survey of over 1,000 people all the way down to a tiny little thief hiding inside your cells. And finally, it gets to a solution that involves, of all things, blue fruit flies.
Okay, you had me at molecular thief, but I am definitely here for blue fruit flies. So what's our mission for this deep dive? What's the big question?
The mission is to finally answer what has just baffled clinicians. Why does the gut fail to heal itself long after the virus is supposedly gone? We're not just looking at symptoms. We're hunting for the actual mechanism, the bug in the software.
I love that. Okay, so let's start with the victims of the crime. This study kicks off with real-world data, right? This isn't just theory.
Not at all. They surveyed 1,032 healthcare workers who'd had COVID. And, these are people who can report symptoms really accurately. The data was... well, it was stark. We're not talking about a stomach bug for a few days.
Right.
We were seeing these gastrointestinal after effects, these sequelae, lasting for 1 to 3 months. In many cases, 3 to 6 months after the infection.
And just to be super clear for everyone, post-infection here means these people are testing negative on those nasal swabs, the standard PCR tests.
Correct. And that's the discrepancy, That's where we have to introduce our first big concept, which is the viral reservoir.
Okay, I've heard this term. Is it sort of like a sleeper cell of the virus?
That's a great way to put it. For a long time, the main theory for long COVID gut issues was that it was systemic, that the whole body was inflamed, like a forest fire that leaves smoke everywhere.
So the gut was just collateral damage in a bigger war.
Exactly. But this paper says, no, wait. They argue for a local intestinal etiology, which basically means the call is coming from inside the house. They found that even when your nose is clear, SARS-CoV-2 viral RNA and antigens, bits of the virus, are still hanging out in your gut tissue.
So the virus clears from the lungs, but it sets up a secret base camp in your intestines.
Precisely. The gut becomes a reservoir. And the virus isn't just sitting there quietly. It's actively sabotaging the local infrastructure. And to get how, you have to picture the gut lining. It's not like the skin on your arm. It's a constant construction site.
Right, because it takes such a beating, it has to constantly replace itself.
It's one of the fastest regenerating tissues in the body.
Right.
The whole thing turns over every few days. And the heroes doing this work are the intestinal stem cells, the ISCs.
The construction crew.
They're the construction crew. Pouring concrete, putting up drywall, fixing potholes, 247.
Okay, so let's stick with that. The city, the gut was bombed during the infection. The bombing's over. Why isn't the crew rebuilding? Are they gone?
They're not gone. They're paralyzed. They're just sitting on the curb looking at all the damage, but they refuse to pick up their tools. And the question is, why?
And this is where the villain of our story comes in.
This is the antagonist. It's the spike protein. Even without the whole virus making copies of itself, just the leftover spike protein in that reservoir acts as a saboteur. But it doesn't use a weapon. It attacks the crew by stealing their lunch.
Stealing their lunch? What do you mean?
It's a metabolic heist.
Yeah.
So the researchers found that the spike protein has a very specific interaction with an essential dietary fat. It's called linoleic acid.
Linoleic acid. That's in things like vegetable oils, nuts, and seeds, right?
Yep. It's essential. Our bodies can't make it. We have to get it from food. Now, get this. The spike protein has this little structural feature, a hydrophobic pocket. The easiest way to picture it is like a baseball glove.
Okay, spike protein is a baseball glove. Got it.
And the linoleic acid molecule is the baseball. The spike protein's glove is perfectly shaped to just snatch that baseball out of the air and hold on to it. It sequesters it.
So it's hoarding all the baseballs. But why does the construction crew care about these baseballs? What's the big deal?
Because that baseball is the key that starts the engine for the whole workday. Inside your stem cells, there's a nuclear receptor called PPAR.
PPA. Okay, let's break that down.
Stands for peroxisome proliferator activated receptor. Forget all that. Just think of PPAR as the foreman of the construction site.
The foreman. Okay.
PPR is the boss who walks onto the site and yells, "All right, time to differentiate you. Go become a useful gut cell." But the thing is, the foreman is asleep in his truck.
And he needs an alarm clock.
He needs an alarm clock. And that signal, the alarm clock, is free-floating linoleic acid.
Oh, I see where this is going.
Normally, linoleic acid comes in, it binds to PPAR, and click, the foreman wakes up, the crew gets to work. But if the spike protein, the glove, is catching all the baseballs,
The alarm never goes off. The foreman just sleeps through his shift.
the construction site stays silent. And here's the really critical part, the deep dive detail. When PPAR, the foreman, doesn't turn on, the cell stops building a very specific piece of machinery, an organelle called the peroxisome.
Okay, stop there. Peroxisome. I feel like I learned about that in high school biology. Isn't that just like the cell's garbage can?
See, that's what they always say. It's such bad branding. It's so much more than a garbage can. It's a highly specialized recycling center. And its main job is to process a really specific kind of toxic waste called VLCFAs.
VLCFA.
Very long-chain fatty acids. Scientists are not creative with names. These things are huge fatty acid chains, like 22 carbons or longer. They're just too big and clunky for the cell's main power plant, the mitochondria, to handle.
So they're like an old sofa that you can't just stuff into the regular incinerator.
Perfect analogy. You can't shove a sofa into a wood chipper. It'll jam the whole machine. You need a specialized industrial shredder to break it down first. The peroxisome is that shredder.
Okay, this is clicking. Let's walk through the logic chain one more time to make sure we've got it.
So do it.
Step 1. Viral reservoir makes spike protein. Step 2. Spike protein, the glove, steals all the linoleic acid baseballs. Step 3. Without the baseballs, the foreman, PPAR, stays asleep. Step 4. Because the foreman's asleep, the cell stops building peroxisomes. The shredders.
Right. And step 5.
Step 5. Without the shredders, the toxic sofas pile up.
You nailed it. The VLCFAs, the sofas, pile up inside the stem cell. And this is the glitch. These high levels of VLCFAs are toxic. They poison the differentiation process.
So the stem cells are literally standing knee-deep in their own garbage.
And because of that toxic environment, they can't do their job. They fail to turn into enterocytes, the cells that absorb water and nutrients.
Yeah.
And they fail to turn into goblet cells, the ones that make protective mucus.
Wow. And that explains the symptoms perfectly. No enterocytes means water just stays in your gut.
Yeah.
Hello, diarrhea. And no goblet cells means no mucus layer to protect the gut. Wow. Hello, pain and inflammation.
Exactly. It explains leaky gut, not as some vague wellness concept, but as a real structural failure. The wall is crumbling because the workers are poisoned by their own trash.
That is a stunningly clear mechanism. But, okay, you can't just say case closed. How did they actually prove this was happening? This is where the Smurfs come in, right?
Yes, the Smurf assay.
Yeah.
Sounds funny, I know, but it's a gold standard test in fruit fly research.
Okay.
The researchers needed a visual way to see if the gut barrier was really broken. So they engineered flies to express the SARS-CoV-2 spike protein just in their gut cells.
So they gave fruit flies long COVID?
In effect, yes. Then they fed all the flies, both normal and the spike protein ones, a bright blue, non-toxic dye. Now, in a healthy fly, the gut is basically a sealed tube.
A straw.
A straw. So if you feed it blue food, you see a neat little blue line right down the middle of its body. The dye stays contained.
Makes sense.
But in the flies, expressing the spike protein, their barrier was compromised. The dye leaked right out of the gut and into the rest of the body cavity.
They turned blue.
They turned completely blue. They became little Smurfs.
That is such a powerful visual. So if the fly is blue, the wall is breached. And did they check why? Did they look for the pileup of toxic sofas in the flies?
They did, and they found the exact same metabolic signature. A severe loss of peroxisomes and a huge accumulation of those toxic VLCFAs.
Okay, but we're not flies. We're not even mice. Did they bring this home to humans?
They did. First in mice, saw the same thing. But the real proof is in human tissue. They got colon biopsies from human long COVID patients, people who were months past their acute infection, and they stained that tissue. looking for a protein called PMP70.
PMP70, what's that?
Think of it as the sign on the door of the recycling plant. It's a protein that sits right on the surface of the peroxisome. If you see lots of PMP70 lighting up, you know the plants are open for business.
And in the healthy people?
The tissue was lit up like a Christmas tree. Lots of PMP70.
Let me guess. In the long COVID patients.
It was dark. The markers were severely depleted. The factories were physically missing from the construction site.
That's the smoking gun. It connects the whole theory, from the baseballs to the blue flies, directly to what's happening in a suffering patient.
It absolutely does. And there's one more thing. Listeners are probably thinking, wait, what about the microbiome? Good bacteria, bad bacteria.
Yeah, you hear about that all the time. Just eat more yogurt, right?
Right. And they did look at it. And yes, they found what we call dysbiosis, a total imbalance.
Yeah.
The good guys like lactobacillus were way down. The bad guys, the opportunistic pathogens, were way up.
So the bacteria are part of the problem.
They are, but, and this is a huge but, the paper argues very convincingly that the dysbiosis is the result, not the cause.
Oh, okay. That flights our usual script.
Totally. Think about our construction site again. The goblet cells aren't making mucus. Well, lactobacillus loves to live in that mucus layer. If you pave over the forest, the animals that live there disappear. The metabolic breakdown poisons the environment, and then the weeds take over.
So just taking probiotics without fixing the peroxisomes is like trying to plant a garden on a concrete slab. You have to fix the soil first.
You have to fix the terrain, fix the metabolism first, and then the healthy microbiome can come back.
This is a huge shift in thinking. It's not about adding good bugs. It's about rebooting the host's machinery, which of course leads to the billion-dollar question, can we fix it? Can we wake up the foreman?
The researchers think we can. Since they found the blockage, the spike protein stealing the keys, they looked for a master key, something that could force PPAR to turn on, even if the linoleic acid was missing.
So a way to bypass the broken lock.
Exactly. They tested 2 drugs that are already FDA approved and on the market for other things. Sodium phenylbutyrate and, much more common one, fenofibrate.
Fenofibrate. Oh wait, isn't that a cholesterol drug? I think my dad takes that.
It's very widely used for lipid disorders. And the reason it works is that, biologically, fenofibrate is a PPAR alpha agonist. That's a fancy way of saying its chemical structure is designed to stick to the foreman and shake him awake. No baseballs required.
Just kicks the door to his office down.
Kicks the door down. And when they gave these drugs to the Smurf flies and the long COVID mice, the results were dramatic.
Tell me.
The PPAR system came roaring back to life. The cells immediately started building new peroxisomes. The shredders came back online. The levels of toxic VLCFAs plummeted. And the most important part, the stem cells started differentiating again.
And the Smurf test. The blue dye.
The blue dye stayed in the gut. The Smurfs turned back into normal flies. The barrier healed itself.
That is just, it's incredibly helpful. We're not talking about some sci-fi molecule that's a decade away. These are drugs sitting on pharmacy shelves right now.
They are. Now, here's my obligatory safety first disclaimer. This is high-grade preclinical evidence. We have the mechanism, we have the animal models, we have the human tissue confirmation of the problem. What we don't have yet is a big randomized human trial for this specific use.
Right. So nobody listening should be running out to self-prescribe cholesterol meds for their stomach.
Please do not do that. These are real drugs with real side effects. But for the clinicians listening, this provides a rational, validated target. It changes the conversation from, gosh, we don't know why you're sick, to, we suspect a very specific metabolic failure, and we actually have tools that might target it.
It gives us a map. Okay, so before we finish, let's just recap the solution side, because that's the big takeaway here.
Okay.
Point 1. The leaky gut seems to be caused by a toxic fat buildup because the cell's recycling centers, the peroxisomes, are shut down.
Right, because the foreman, PPAR, is asleep.
Point 2. That happens because the spike protein steals the on switch, the linoleic acid.
Yep, the baseballs are gone.
Point 3. Drugs like fenofibrate can act as a master key, a manual override, to turn the whole system back on.
And point 4, in the lab models, this cleared out the toxic waste and allowed the gut wall to physically heal itself.
A perfect summary. Okay, so for our final thought, let's zoom out. We've been focused entirely on the gut. But the spike protein, it doesn't just stay there, does it?
It doesn't. And this is the thought that I can't shake. We have this clear mechanism now. Spike steals a fatty acid, which break peroxisomes, which leads to toxic buildup and organ dysfunction.
Right.
Well, the gut isn't the only organ in the body that is incredibly dependent on fat metabolism.
Where are you going with this? The brain.
the brain. It's about 60% fat. It's one of those metabolically active organs we have. And we know from rare genetic diseases that when peroxisomes fail in a person's brain, it causes devastating neurological symptoms.
You're talking about brain fog.
What if brain fog isn't just some vague feeling of being tired? What if the spike protein is performing the exact same fatty acid heist in the support cells of the brain, stealing the fuel, shutting down the shredders, causing a toxic buildup? Maybe brain fog is just leaky brain.
It's the same glitch in a different machine?
It's a hypothesis, but it's a powerful one. It suggests that maybe long COVID, at its very core, is a systemic metabolic disorder triggered by these viral remnants. And if that's true, then metabolic therapies fixing how our cells handle energy and waste might be the key to the whole syndrome.
That's a huge idea. That shifts the whole paradigm from fighting a virus to repairing the metabolism.
And that gives us a whole new toolbox.
Well, listeners, that is a lot to digest. We really want to hear from you on this one. Does this mechanism resonate? Have you noticed your own symptoms changing with, say, high-fat or low-fat meals? And for any clinicians listening, does this change how you might think about these incredibly persistent post-viral symptoms?
We'd love to hear if there's any anecdotal evidence out there that lines up with this fatty acid thief idea.
The paper, again, is Impaired VLCFA-peroxisome-mediated intestinal epithelial repair causes gastrointestinal sequelae of long COVID. It's a mouthful, but it might just be the answer key we've all been waiting for.
It's definitely a light in the dark.
Thanks for diving deep with us today. Take care of your peroxisomes. We'll catch you on the next one.
Stay curious.